Welcome and thank you for joining today's conference, CWD Program Standards 2018. Please note that all participant lines will be muted until the Q&A portion of the call. We'll provide you with instructions to ask a verbal question at that time. You are welcome to submit written questions during the presentation, and these will be addressed during Q&A. To submit a written question, use the chat panel on the right-hand side of your screen. Choose All Panelists from the Send To drop-down menu. If you require technical assistance, send a note to the event producer. With that, I'll turn the call over to Dr. Alicia Noggle. Please go ahead.
Thank you. Good afternoon, everyone. My name is Alicia Noggle, and I am the director for the Sheep, Goat, Cervid, and Equine Health Center in APHIS's Veterinary Services. And one of the teams that's under my direction is the Cervid Health team. And we're so pleased that you're joining us today for us to provide an update on our revised chronic wasting disease CWD program standards. Today's call is targeted toward a really important stakeholder group for our herd certification program, accredited veterinarians and sample collectors. So we really appreciate you taking time out of your busy schedules to join us this afternoon. We have a couple of purposes for our time together this afternoon. First, we plan to provide several highlights of the changes that we've made to the program standards from the prior version. Secondly, we want to use this time to really answer your questions that you may have about these changes and clarify anything that perhaps isn't clear in the standards that we need to revise moving forward. Again, I will remind you that we've really tailored our conversation for this afternoon for you, and we really rely on your help for the fidelity and the program, the importance of the program, because without those good, high-quality surveillance samples, we just don't have the program integrity that we need. So we really appreciate your hard work. The last thing I'll share with you is we are during a comment period. So we're happy to take written comments. In fact, if you have comments, we prefer to receive them through the website rather than today. That ensures that we'll address them in a systematic way and we don't lose them in the conversation today. Those comments can be submitted to www.regulations.gov through April 30th. So with that, I'm going to turn it over to two of my team members to share some of the changes in the program standards with you, Dr. Tracy Nichols and Dr. Nancy Hanaway. So Dr. Nichols, I'll turn the floor to you.
And I'm going to turn it right over to Dr. Hanaway. Thank you, Tracy. She's going to start us off today.
Okay, some content changes involving sampling. We're going to go over consequences of poor quality and missing samples. Next, we'll go over sample collection and submission. Then we'll go on to anti-mortem diagnostics. And finally, biosecurity recommendations for farm service facilities. First, we're going to talk about the consequences of poor quality and missing samples. As Dr. Noggle said, this is critical to the integrity of our program, and surveillance is the key to increasing our confidence that HCP-certified herds are at low risk for CWD. So it's very important that we get the surveillance that is needed in these herds. In the new program standards, we have listed consequences for participating states to implement in response to this issue. Missing and poor quality samples, like we said, will result in consequences of the herd's owner's HCP status. So please don't let that be your fault. Some of the consequences may include that they'll need to replace missed or poor quality samples with samples from an equal number of animals of the same sex and species that resided in the herd as long or at least as long as the missed animal. Another consequence will be reduced herd status, either a loss, a reduction, or delay in certification. And finally, another consequence could be direct suspension of the herd status for a designated period of time. And like I said, that is up to the states to impose those consequences.
Okay, this is Tracy, and I'm going to talk about a number of things regarding sample collection. I'm going to start off with slaughter surveillance. So it became obvious that our language was not consistent on some items between the CFR and the program standards, and one of those items was slaughter surveillance. So we revised the language of the program standards to reflect the requirement that's in the CFR. So this is not new, it just wasn't included in the previous CFR. And that is that all mortalities, including animals taken to slaughter under HCP participant ownership, must be available for CWD tissue sampling. So if they're owned by the same person and they go to slaughter and they're part of our HCP, those animals need to be tested. So let's talk about sample collection. This is an issue that's been brought to our attention that appears to be kind of a problem sort of across the board. And I want to reinforce this. Now, this was in our previous program standards, but I feel like it really needs to be emphasized, and everyone needs a good reminder about this. Samples should be submitted within 7 days of collection. 7 days. Even faster is better. Okay, so no riding around in trucks because, you know, you haven't quite got to it. That's a problem, and hoarding hurts, and here's why. So let's say you've taken a sample from an animal, a mortality on a site, and it doesn't get in within that 7-day window. It sits around for a couple weeks. During that time, that owner may have decided to sell some animals to someone else. So when we get the results from that sample that finally ends up getting turned in, oh my gosh, it's positive. That means more owners have now been drawn into a trace-out situation that never should have been had we detected this early and there hadn't been a sale or movement of animals. So please, hoarding hurts. Please get those samples submitted within 7 days. Just a reminder, please take care with sample ID. This year there's been some issues with sample ID mix-ups or the wrong sample being in a vial. This really is not something that should be left to technicians or someone that doesn't understand the gravity of the situation. This could mean, if it's incorrectly sampled, that you could be putting an innocent producer under quarantine and potentially out of business through this kind of mistake. So when I go on sites and I do sample collection, I always tell the person who's writing down the numbers that they are the most important person on the site, because incorrect sample IDs are devastating potentially. So please take care with that. And again, this goes right into that. Carefully and accurately label samples. Sample collection. So there are new requirements for fixed and fresh post-mortem regulatory samples. So this is important, and I'll tell you why we've made some of these changes. So for fixed, of course, we still want the OVEX. So you're going to take and put the actual OVEX itself into 10% formalin, clearly and appropriately labeled. But what we're asking you to do in addition are those trimmed off pieces of brainstem to go ahead and drop them into a whirl pack. Okay? So before you may have put the whole thing in, or I think a lot of people do actually trim and then they just kind of get rid of those additional brainstem trimmings, please go ahead and save those. And you're going to be asked to save some other things as well here. Okay, the brainstem here we're talking about, putting those into whirl packs, so I've kind of already covered that. The next change is going to be in regard to medial retropharyngeal lymph nodes. So this is our other regulatory tissue. And we're asking that you longitudinally cut the left and the right medial retropharyngeal lymph node, and that you place half of each in the formalin jar. Okay? The reason is that in a percentage of cases, there's unilateral distribution of CWD. So if we're lucky, the one lymph node that is submitted is the one that's positive, but what about the one on the other side? So this change was implemented to make sure we are not missing unilateral cases in medial retropharyngeals. So what about those other halves? We're going to ask that you take the other halves and place those in the whirl pack with your brainstem trimmings and submit those fresh. And the reason we want to do this is kind of twofold. First, this provides us some extra test tissue. If there's a question of ID, then we have some fresh samples with DNA, and that allows us to check on that. And secondly, it does provide a tissue if we decide we want to do some genetics regarding Codon 96. Sometimes that's very informative for epidemiological investigations. If we do not have fresh material, we cannot do genetics. And that's because formalin forms disulfide bonds with the DNA, and then you can't amplify it. So it doesn't take too terribly long, and that's another reason to get samples in timely fashion, is that in about 2 weeks or so, getting DNA from fixed tissues becomes downright impossible. So we need that, because there have been situations where we need to identify individuals and we need some fresh tissue, or if we need to double check, then we've got it. A new sample that we're asking that you collect, and this is another important one for appropriate identification if there's a question, is the official ID with a piece of skin attached. So about a 1 inch by 1 inch piece of tissue with the official ID attached. Go ahead and drop that into the World Pack with your brainstem trimmings and your medial retropharyngeal extras. This should not go into formalin. Now, I've seen this happen where folks are, cool, I'm going to do the right thing. I'm going to sample an ID tag, and I'm going to submit that. However, they drop it into formalin. And for those reasons that I stated in the previous slide with regard to the integrity of the DNA, that's not what we kind of need you to do. We need you to submit that fresh. And this goes back to the need to have official ID with tissue if DNA ID matching is required. So this is very important. So you're going to submit those right along with your formalin fix. You're going to send that little package of those things in. Of course, just a reminder, on your sample labeling, you want the date of collection, the producer name, species, type of specimen, official ID, and sample ID number. So what is assigned on the NBSL 10-4 form. That's a good idea, too, if you get a chance to do that. So let's talk about Mortem Diagnostics. So this is new to the program standards. We've been talking about this for quite a long time, and we finally got this implemented and into the new program standards. So we're going to get into the weeds a little bit about how this is going to work, because it's not immediately apparent. I think we need to get into it. So who can be sampled? At this point, it's white-tailed deer only. Why is that? Because we don't have sufficient data for elk or mule deer to do the same kind of calculations for detection that we have done with white-tailed deer. That doesn't mean that that couldn't happen in the future. That just means at this current time, we don't have the necessary data. So who can collect the Mortem Diagnostics? And this is going to be an important point to keep in mind. It has to be done by an accredited veterinarian. So unfortunately, sample collectors, your official sample collectors out there, this is not going to be something you can do, because this is surgery. This has to be done by a veterinarian. In addition, the collection by accredited veterinarians must be supervised by a state animal health or VS representative. So this isn't one that you can do on your own. There's accredited veterinarians, and there's a reason, and we're going to tie this into teaming up with a herd plan, which is what this kind of comes down to. So what type of Mortem diagnostics are we talking about? We're talking about rectal biopsy and medial retropharyngeal biopsy via immunohistochemistry, so IHC. So what is the deal with the MRPLN biopsy? And that may be a new one to a lot of you folks. A couple years ago, there was a group of veterinarians in Texas that had developed a way of biopsying medial retropharyngeals. Not an easy task, I would say. That was kind of a hot topic for a while. We haven't heard much about it lately, but during that time, we worked with NBSL to sort of, excuse me, develop parameters around that. So we'll get into those parameters here in a second, but that's what that is if you haven't heard that before. Here's some important aspects to the biopsies. More than one test is required. This is not a one-and-done test. So this is because of the lag period of CWD incubation. One shot is not going to be enough, because we're testing with the rectal biopsies the back of the animal. It has to have time to get there. This is also a whole herd test. Because of our probability of being able to detect that one individual that's positive, it's got to be whole herd. So this is not an individual test at all. This isn't a way for one animal to get approval to move or something like that. That is not how this works. So what are the conditions in which this is going to be employed? Where is this happening? This must be outlined in the herd plan. So the testing timeline, when I mentioned there's more than one, this is also going to be established in the herd plan. So this is not something that's going to happen just whenever or wherever. This is under very select and qualified circumstances. So why did we decide to move in this direction? Because we wanted to assist producers drawn to quarantine situations to be able to restore business continuity faster. 5 years, you know, is a pretty reasonably long period of time. So we were trying to figure out a way to assist those individuals that just had the situation where they may have sold an animal to a positive site within the last 5 years. They're drawn into a quarantine. How can we get those folks out faster and have reliable information? So when will we use this? It would be implemented for quarantine situations on traceback herds or CWD exposed herds or epi-linked herds. So very select set of circumstances, not for routine surveillance. That's an important take-home aspect to this. So when is this considered appropriate? First of all, we need to understand the genotype at codon 96 because it impacts the progression of the disease. So first of all, we've got to genotype the herd. We need over a 50% GG ratio, so this is our wild type, most rapidly progressing genotype. It needs to be over 50% to utilize the rectal biopsy. Excuse me. That's because when we looked at probability calculations, we get over 50% GG, and we get into some of the slower progressing animals, our ability to detect animals goes way down. Okay, and that's because this influences incubation period. So just as a reminder, GG animals progress more quickly than GS animals, and GS animals progress more quickly than SS. So we're looking at a biopsy at the back of the animal. So just as a review, the disease comes in the front of the animal, it proliferates in the regional lymph nodes, and then it makes its way back to the lymphoid tissues in the rectum, and then finally it makes its way back up to the brain in white-tailed deer. So there's a lag period in there that we're trying to work around and still have accurate results. So how is this to be done? Inside the new program standards, we have provided very specific collection and submission guidance. So that's in there, so you'll be able to refer to that. What makes these samples a little different is the testing must be done via NDSL. So you cannot send these antemortem samples to other non-laboratories. And it's going to be at the owner's expense. And the type of assay used on the biopsies is going to be IHC. So what are we looking for specifics-wise on these biopsies? So for rectal biopsy, we're looking for a biopsy of about 1 centimeter by 1.5 centimeters. And as most of us are aware, the number of follicles present needs to be greater than 6 in order to be an acceptable sample. Any less than this, and you'll get your official report back that says ISF for insufficient follicles. With the medial rectopharyngeal biopsy, the amount of tissue needed was established by NVSL, and it was compared to post-mortem. So the amount was 2 centimeters by 1 centimeter by 1 centimeter, and there needs to be at least 40 follicles included in that. Also, with the blood, that needs to be collected fresh, obviously, 3 to 5 mils of whole blood in EDTA to check for codon 96 genetics. It can be chilled or frozen, and GeneCheck currently seems to be our most appropriate lab to do this, as they're approved to do scrapie testing, and they have clear-cut quality control and quality assurance policies in place for that type of testing. Again, samples for this should be submitted within 7 days of collection. The rectal or medial retropharyngeal lymph node biopsies, you're going to place those in the 10% formalin jar that's clearly labeled. And the rectal biopsy itself is going to go in a cassette. And what's missing from this image is the top piece of foam. That's important because the rectal biopsy samples will curl in formalin very quickly. And without that top piece of foam to hold it down, it'll curl even in the cassette. So those are going to be placed mucosal side down on the cassette, another piece of foam on top, and then dropped into the labeled formalin jar. Okay. So that's kind of the high-level view of the sample collection. Another aspect that we thought that you all should be aware of are our biosecurity recommendations, and this is kind of important because you all may be going to numerous farms. So just like other diseases, we want you all and our producers to start thinking about biosecurity and what you're doing and what you're wearing to and from sites. So, you know, biosecurity are procedures to reduce the risk of introducing CWD or other diseases to a farm. So some of the things that we've recommended to producers to start trying to handle are direct contact between cervids and wildlife. Of course, direct contact between captives and wild is in no one's best interest, so if that can be reduced, we've got some recommendations there regarding moving feed away from the fence line, reducing forage along the fence line, things like that to make the fence line less attractive to both sides. Measures to reduce bird problems. That's kind of a big problem with some producers, some more than others, but figuring out how to keep birds out of their feeders and waterers. Feral hogs, those guys can bring any number of things onto a farm. We don't know what their role is with CWD currently, but they are not customers you want eating at your deer food diner. And then, of course, other animals are best kept elsewhere as well. Probably what's more relevant to you folks is the prevention of indirect contact with fomites, so potentially contaminated objects or material. We are encouraging them not to store their feed in hay outside of the fence where wild deer could come and feed on it, and then they're going to throw it in their pens. That's just not a good idea. And human traffic, and that comes down to folks that are coming on and off. If you're wearing your boots from one deer farm to the next, this is probably not a particularly good idea. Shoe covers would be a great thing. We're also encouraging producers to perhaps buy some specific boots for their farm that when they have visitors coming to their farm, either to help them run deer through or to help them medicate or sample or AI, you name it, to have those available for people to slip into, leave there, and then leave. So you're not bringing your boots and your equipment that you've just worked on the farm down the road into another farm. This is just kind of good common sense, regardless of the type of disease we're talking about. And we also recommend that they start thinking about their vehicles and equipment and not moving those around. So at this time,